Welcome to the PharmaSource podcast. Today's interview is with Herman Bozenhardt, a consultant in biopharma manufacturing with decades of experience in facility design and operations, having worked with numerous pharma companies and contract manufacturers. In this episode, Herman shares some pretty candid thoughts on the challenges facing manufacturing, including his view on why, in the rapidly growing field of antibody-drug conjugates, ADCs, many CDMOs may not be adequately prepared to handle these complex and potent compounds. I hope you enjoy the episode. Herman, thank you very much for joining us. Welcome to the PharmaSource podcast.
It's a pleasure to be here.
So you write a lot, you write extensively with your son, Eric.
Mm-hmm.
So in putting this conversation together, there's plenty of material out there for us to talk about. So what I thought we could do is have a sort of a warts and all conversation about some of the issues, some of the challenges around manufacturing, particularly with contract manufacturing. I thought we could start off talking about ADCs. Now, it's a topic which is a really big growth area at the moment. Every day there's some news about ADCs, but I think you're a bit more skeptical about ADCs from what I can tell. What are your thoughts on the category?
I'm very, very positive about them in terms of an avenue of therapy.
Sure.
There's no question about it. It's a targeted area with a good payload on it, whatever. And if the processes and facilities are built from scratch, they're designed properly, no problem at all. But what are the pitfalls that we have in our industry today is that we have a lot of CMOs and CDMOs who are going ahead and say, okay, we'll take it on. Yeah, sure. We'll help you, Mr. R&D type pharmaceutical company. We're going to bring it in here and we're going to do it. But do they really know what they're getting into? And does the actual R&D end of the company know what they're getting into?
Because there are a lot of CDMOs at the moment who are promoting themselves as being ADC specialists.
That's exactly right. And the real question is, do they know what they're talking about? Or they're just trying to attract business? The big fallacy is just like anything else. Whether you're a salesman for the CMO or you're selling a used Volvo from 1984 Volvo, there's no difference. Oh, sure, we can do that. No problem. It'll be fine. Great. We're good. The real issue is handling the potent compound.
Yeah.
And the process is no problem. It's not that difficult of a process. We can always conquer that. We can get that together. But bringing in a highly potent compound. Now, we're not talking about the typical brands, band C and Ds. We're talking about people now bringing in E and F bands with very, very critical OELs. And now how are we going to be bringing it into the facility? How are we going to be handling it? Oh, well, we're going to buy an isolator. Oh, that's great. We're going to buy an isolator. But how did you bring that bulk material in? Did you bring it through the warehouse? Did you bring it through the hallways? Where did you store it before? Did you store it in your regular warehouse? But what happens if a forklift should spear it, or the crane that's dealing with the material should drop it and crack the canister? Then your warehouse is all contaminated, and you can't use anything in the warehouse. Then when you bring it in, we're going to be processing it. And now how do we process it? How do you clean that? And I'm going to use the word clean in two segments. How do you pick up if you have a spill? How do you clean the inside of an isolator? But more importantly, how do you neutralize it? See, everybody says, well, geez, Herman, it should be no problem. We're going to go wipe down the inside of the insulator with some WFI. Then we're going to wipe it down with some alcohol. We're good to go, right? Well, no. If you're going to wipe it with alcohol, with water first, all you're going to do is dilute it. It's still potent. And when it dries, it's still potent. I'm going to wipe it down with alcohol. Well, alcohol is going to make sure it's microbial free, but it's still potent. You haven't changed anything. So that isolator is contaminated now. When you open it, when you close it, whatever. We have the air on it all the time. Well, that's great, but when it dries on the surface, so we have to make sure that we have a material that will neutralize it to completely inert, and then we can remove that. Then we can worry about microbial contamination. But the whole issue of neutralization is the big issue.
Your sense, Herman, is that isn't happening in many cases.
That's exactly right. The concept of neutralization the concept of how do we handle it in the isolator, because I'll go out on a limb and say I wouldn't, if I was handling a highly potent compound like this, I wouldn't buy a fixed stainless steel isolator. I would buy a disposable isolator.
Okay.
I use it once, I do my manipulations, I'm all done, and then I carefully collapse it, put it in a plastic bag, and send it on out.
Hmm.
Send it to an
Hmm.
incinerator, as opposed to having to worry about not only cleaning it, but then I have to develop an analytical method that's going to be good enough so I could swab the inside to make sure that I don't have it, it's not contaminated. I also have to make sure I have to swab the people who are working in the area, have to swab the facility, the suite that I'm working in, to make sure I don't have any contamination.
What's the FDA's view on this? Are they looking at it in a similar way to you?
Yes, they have to.
Hmm.
The fact of the matter is that whether we're dealing with ADCs or we're dealing with testosterone, there was years ago, there was a big controversy with a facility that was handling testosterone and filling in the suite right next to one that is handling some other injectable. And it's a
Hmm.
highly potent compound. We deal with a lot of the chemotherapeutics in our world, or I should say, the payload for liposomes for different oncology therapies.
Hmm.
In the same way, the FDA asks, well, how are you handling this? Where is it being handled? What did you do for manipulation, moving it in and out? The only thing is, with the ADCs, we are getting into higher and higher risk categories because of the
Hmm.
lower OELs. Our OELs now are ferociously low. So we are we're increasing our risk levels. So now how do we handle potential cross-contamination? How do we protect our employees? And the FDA is very interested in how we worry about cross-contamination. Do you have the facility to do it? Do you have the HVAC correct?
Hmm.
So you have bubble sink on the airlocks to make sure you get everything contained. Then you
So,
ask the question. Go ahead. So
no,
then you
no,
ask
please.
the question, where we're running, everything's going good. What happens if you have power outage? Whoops. Well, and the containment is by keeping the air pulled in. Well, do we have an HVAC that's ready and keeping that at area negative? Okay, if it's
Hmm.
negative, well, that's great. But do do we have a battery backup? Do we have a backup diesel generator? What if when the HVAC goes down or power outage goes down, how long does it take before the backup generator turns on? If you're telling me, 1 to 5 minutes, so you have 1 to 5 minutes of potential um, uh, egress of the potent compound. Can't
Hmm.
do that. You have to have a battery backup on the HVAC. So there's a lot of issues associated with facility design that
Hmm.
I don't
Hmm.
think is really people are really understanding.
Yeah, yeah.
Secondly, is let's be frank about the CMO business. CMO business is, we buy old beat up facilities from whatever they were, whether from Glaxo or Merck or Bristol Myers, and we go ahead and we retrofit them, and we start a business.
Hmm.
And so it's more than just the suites and the SOPs. It's the HVAC system. It's the warehouse. It's the path in, the path out. And so we don't necessarily have an infrastructure and a facility design model that really fits something like the ADCs. Now,
Hmm.
maybe there's ADCs out there that have started from scratch, brand new greenfield, and they built them up. I don't know of any. Okay.
Hmm.
Maybe there are some. Some people tell me that they are. I don't know.
How many would you say that there are out of the, let's say, 80 or so, for sake of argument, 80 CDMOs who are saying that they're doing ADC services right now? How many would you say, in your opinion, would probably be fit for purpose?
Probably five.
Wow. Okay. So very few.
Yeah, very, very few. Because what typical is, and I'm familiar with plenty of the CMOs. I know exactly how they work. They say, yes, Luke, fine, great, we'll do it for you. And then they run back and they tell operations, hey, operations, you have to do this. And they're like, oh, wait a minute, what do we have to do?
Hmm.
The whole point is to get the business, bring it in, book it, and then worry about it later. Let somebody else worry about it.
So if you were advising an ADC developer, a biotech or a small pharma company who's developing a new ADC drug product, what advice would you give them as to outsourcing their manufacturing?
First thing I would do is I would go with organizations that are reasonably large and have a decent sized facilities and have some cash behind them that can
Mhm,
invest in doing the work for you. That's the first thing.
mhm,
Secondly, is I would take very, very seriously there's guidelines out there. In fact, there's some papers that my son and I have written about high potent compounds, and look at that and do the risk assessment. It's from the American Institute of Chemical Engineers. It's from ISPE best practices and guidelines.
mhm,
And look at that and look at the risk profile and understand really fully what is required in a facility design. And then go ahead and look at several CMOs and audit them against those criteria. And
mhm,
when they say to you things like, "Oh, don't worry about that. We'll build that for you," or "Don't worry about that. We'll take care of that," you got to have something in the contract that says this is what we're going to do. Because we've had situations where, even for non-potent compounds, where the CMO tells us, "Oh, don't worry about. We have a traditional filling line here with some raggedy ass curtains. We're going to put a brand new isolator in for you." And so
mhm,
4 years later, guess what? There's no isolator. "Oh, we'll be okay. We'll finish up these couple of batches and then we'll move it here, move it there, and it'll be fine."
mhm.
Well, the whole issue has to be with, commitments and what's in the contract. A lot of organizations, yeah, especially the ADC developers, are not organizations with huge, deep pockets,
Mhm.
and they need to buddy up with or work with somebody who has deep pockets, who looks at this and says, "Well, if I do an ADC for them, I may be able to attract some other business with ADC. So I will build a new facility. I will buy modular systems from Germ-Free or GCon or some of the other module, and we'll put them in, we'll stack them up in this shell building, and we'll get everything right."
Mhm, mhm.
So you cannot have a small company and another small company, because I don't think there's going to be enough critical mass of cash to get it correct. So again, understand your risks, understand what you're working with, audit against that, and make sure that's in the contract.
Yeah. I mean, give it if it's the case that there's only, let's say, five CDMOs who are actually fit for purpose for handling these kind of highly potent materials, that's going to cause quite a bottleneck because there's so much in the pipeline in these kind of modalities.
That's exactly right. And I just see this as a huge bottleneck.
Mm,
I see the big pharma who's got the deep pockets and the facilities. They will leapfrog everybody else.
yeah.
And so what does that mean? Maybe it means that they're going to have to partner. They're going to have to partner with one of the big pharmas and work with them. Um, unless they find somebody who sees this piece of business and doing the CDMO for one of these developers as a step into the business. Everybody
Mm,
can say they handle potent compounds. It's easy, you know, but doing it correctly. I mean, if you go back, let's look at, I hate to go, I'll go to the biological side of the business, and you can go look at the 2001 April 483 for the emergent biosciences. And you look at emergent biosciences, which is a very reasonably reputable um, CDMO or
mhm,
CMO for manufacturing, and they took on business from J&J and AstraZeneca. Okay, and you would figure big facility, and you read the 483, and it showed about the issues of cross-contamination, lack of separation between the two product lines, and all. I mean that 483 was huge,
mm,
and I think that that's changed the whole nature of the COVID vaccine back in 2000 and 2001 by basically taking them out of the business. I'm not saying, and all I'm basically referring to is what I've seen in the 483, which is a public business. So we think, here's a big company, they would know how to do it. Well, it
mm.
looks like they didn't. You would
Yeah.
read that, and my opinion is by reading that 43, it looks like, geez, they really didn't know what they were doing. So it's easy to take business. When you see the dollars, you'll take it in. But the practicality of going ahead and operating that way, because you're
Mm.
dealing with a very high potent compound, the people are going to have to be gowned up several times. They're going to have to put an additional suit on. They're going to have to be sprayed down as they come on out, especially if they're handling that material. Then they're going to have to discard the suits. They're going to have to take a shower and then move on out. Are they
Mm.
doing it all the time? Is it being done correctly? Are they isolating all the tools and the instruments that are inside? So there's besides the facility and that, it's also the understanding the practices that got to go ahead. It's
Yeah.
not that easy. And
Yeah.
you can see what can or what cannot happen.
So what are some of those horror stories that you've, I mean, not just around necessarily ADCs and high potent compounds, but just generally around facilities. You've written in the past about uh finding finding pigeons in a plant
Oh
or
yeah.
or cats in a warehouse. Like what kind of examples have you got?
Well, for example, I'll give you the pigeons in the plant. So there was a facility that remains nameless. It's in the United States, and they had asked me to come in and take a look at the facility.
Mm.
And because there were going to be a change out of the FDA inspectors, they were concerned about where that particular facility is going to be going in the future. So I went in the facility, and it's a big API facility, big reactors, kind of a, it's all a gray space type environment. At some level, yes, but at it was what I would consider dirty, filthy.
Hmm.
Um, and I'm walking around there, and I keep seeing all these white spots all over the tops of the vessels. And I'm like, you guys are pretty sloppy with your transfer. And you have the hatch over there. Did you have something flow over or whatever? Oh, no, no, that's the pigeon droppings. Pigeon droppings? I said, well, well, it's a long story, Herman, pigeons got in and whatever. Okay. So
Mm.
I'm going along, and I said, you cannot have pigeons inside a pharmaceutical plant. I mean, that's in case, anybody's listening, go to realize that. So we're walking along, and I saw they had what they called a sterile or clean subdivision table. And it
Mm.
was a stainless steel table with laminar flow hoods above it, but still in this dirty, filthy environment. And I said, how do you justify that without any kind of airlocks or anything like that around it? Well, Herman, it's just a bulk, but yeah, it's a bulk that could become contaminated, and then you have the microorganisms, and they'll be throwing off some endotoxin. Oh yeah, we shouldn't have that. And oh, by the
Mm.
way, look at this right here, not too far from there, is this filthy, jalousie, rusted window. And if somebody may not know what a jalousie window is, it's those windows where the glass panes crank out.
Mm.
And I said, you still have the jalousie windows here. There's no screens. And the guy says, I said, you don't use that. And he says, oh yeah, we use it in the summertime when it gets too hot in here. I said, but you
Mm.
don't have any screens on that. And he says, well, that's how the pigeon got in.
Yeah.
So,
Takes us back to the
there,
pigeons.
yeah. So
Yeah.
we've seen that, and we've seen the facility in the U.S. also where we're going in the warehouse. And it's kind of an interesting phenomenon. You're walking along, you're talking to people, you're writing stuff down, looking at a GMP finished goods warehouse, and all of a sudden see like a flash, like right on the peripheral of your vision. It's like,
Mm.
is that me? I don't know, maybe. And then I was like, maybe it's just me. And I'm walking along, I see a cat, a white and orange cat jump from pallet to pallet.
Mm.
And I'm like, what's that? And the guy says to me, it's a cat.
Did
And
the
then
cat have a name or was it a stray?
no, I said, what's a cat doing inside a GMP finished goods warehouse?
Mm.
And the gentleman says to me, it's part of our pest control practice. So you
Y-
have live animals chasing other live animals inside your warehouse.
Yeah.
And he said, well, if you want to put it that way, So then later on, as the guys are walking, that somebody who was with me all of a sudden tripped, and they tripped on a litter box. And I said, you have feces and urine inside the GMP warehouse. Oh, he said, that's for the cats. So when they catch the mice, so I said,
But it
look,
sounds like the cat's part of the quality oversight plan.
that's exactly right. Yeah, in fact, I think the cat was the head of quality in that particular.
Yeah.
But the reality is look, Luke. I grew up in Brooklyn, in a crummy area in Brooklyn in New York City. My facility that I first worked in was Pfizer in Brooklyn in
Hmm.
Bed-Stuy. Even though Bed-Stuy was not a great place, we had a fabulous warehouse. We never had a pest problem. We had multiple doors and locks and pest control systems and traps and the blue lights and everything. We never had a problem.
Hmm.
And all of a sudden, these guys are telling me that, well, the reality of being inside of an inner city is just too tough for them. They have to have live cats. So, I've seen just about everything.
Yeah. Yeah.
I've seen everything. I've seen, and one of the things and you look at our world today, we have a lack of appreciation of BL1, BL2, even BL3.
Hmm.
And in some of the CMOs, in some of the older facilities, we do not also have a good and a proper appreciation or proper understanding of pre-viral versus post-viral.
Hmm.
Now, I've written about this before. If you look at, one of the most prettiest and most well-designed facilities for, the 2000 time frame was J&J's Malvern facility. That was a beautiful facility. I mean, essentially, the pre and the post-viral was separate buildings, which was great.
Hmm.
But I see facilities today where people are interacting pre-viral and post-viral. They're interchanging. Pre-viral people are walking in and doing work in the post-viral. You can't do that.
Hmm.
You've had a microorganism working over here. You've had some kind of a product that's pre-viral clearance, and then they go ahead and they have to help out in the post-viral where they have done the sterile formulation. Well, no, you can't really do that. And so they look at that and uh, there's not a good understanding. So we
Yeah.
see all sorts of stuff, a wide spectrum. The problem is that we as an industry have organizations that have facilities that were built in the 70s and the 80s. As times
Hmm.
have changed,
Yeah.
regulations have changed. In our understanding of products and contamination have changed. Look at HIV. HIV didn't come till the mid 80s. Um, we we don't these some of these facilities are not maintained very, very well. The products that they have put in them are not the original products. So we've gone through, several layers of modification, not I should say several layers of change, regulations, products, uh, criticality of the products, type of products. And now we're sitting in a facility that was never really geared for that. And we're trying to stuff them in there and try to make it work.
Yeah.
And there is where our problem always rises from.
Hmm. Hmm.
This is where our danger is. And
What?
so
What?
we've either got to face the situation or we've got to move on.
Yeah. So for a manufacturer using multiple smaller process trains
Yeah.
versus having single large-scale bioreactors, what's your take on that, and what are the benefits of those approaches?
See, there's a couple of pieces of that.
Hmm.
All has to do with risk assessment. We are getting to the point where we have learned and we have evolved. We have learned about the titers and the potencies of our products, and we've learned how to put them in pilot plants, run them under automation, and we realize, oh, wow, we can get a lot higher density. Okay. Um, Now, do we put all, and then we're going to run in a bioreactor, say 5,000 liters. We're going to put all the company's money, all our products in all one shot. We could have a contamination, especially if it's a stainless steel reactor. We
Hmm.
could, the CIP, whatever, we could have a potential problem in there, and then we could lose everything. So I always advocate is to either go to single-use disposables completely and go to smaller sizes so to lower our risk. And the risk not only is not getting the product to market, but it's also your insurance risk. There was
Mhm.
I did some work for an organization several years ago, 10, 15 years ago, where we were going to be building this massive lyo that was going to take, it's going to basically do a couple of months worth of product for this particular company and going to give them all sorts of capacity. And then when we putting it together, the risk management and insurance group actually came back to us and said, you can't put all that in one lyo. The insurance company will not cover us if we have a fault in there, we have a contamination. They're not going to cover us.
Mm.
So there's another aspect of risk is, if we've put this particular critical therapy product together and we have it and we lose it, what is the consequences not only from the patients on the market, but what is the consequences from dollars and cents? Are we covered? And
Yeah.
interestingly enough, is over the years we have found that insurance companies are not willing to cover that. So there's both financial risk as well as therapy risk. So what I advocate is looking at the dollars and cents, looking at the potential for failures, and lower your risk. We
Mm.
built years ago 40,000 liter bioreactors.
Yeah.
Terrific, wonderful technology. But guess what happens if you lose the store? So we've got to be a little bit more practical. Our value of our products are going up. The potencies are going up in terms of not needing as big of vessels such as that,
Mhm.
and the overall risk in our manufacturing space. So typically, I prefer people to look at it. If you're making a commodity product, you're making saline solution, glucose solution, sure. If
Mm.
you if you if you you know you screw up a batch of saline, you flush it down the drain, no big deal, not a big loss. You refill, you go on from there, you solve the problem first, and then you go ahead and you can remanufacture. But if you're dealing with a monoclonal antibody, or you're dealing with a critical biological, you got to say to yourself, well, what the heck am I doing?
Mm.
And what is my risk associated with that? And when you see a loss, when you see you've put a big lyo and the lyo shelf control system makes a mistake and you crush a whole series of vials and you have glass all through the lyo and you have to say to yourself, well, wait a minute, that all that has to go. And then you realize, wait a minute, we'll need another X number of months before we get all that material together. Years ago, I did a product for another one of my clients. It's a 17-day process. And
Mm.
I told the operator, I said, we got to be very careful what we do because we could be perfect all the way through. And on day 16, we make a mistake. We have to throw this all out. Now we have to throw it all out. We have to get all the raw materials together. We have to get all the single-use disposables together. And then we have to find another opening in our manufacturing window.
Mm.
So a miss on day 16 could delay us months. And these products aren't going to go to the client or to the patient. So as we get to more and more critical therapies, our risks are not only to ourselves, but to our patients.
So, I mean, all of this sounds like big levels of investment. Where do other types of facility upgrades fit in? So, thinking about manufacturers who want to implement digital technology, bring in digitization and AI into the manufacturing, or on the topic of decarbonization, how they can create more sustainable ways of doing this. It's a huge amount of investment, which most manufacturers aren't going to be able to cover, I presume.
Well, one of the things I'm going to say about AI, because I did a lot of AI back in the very, very early stages of AI.
I saw 1988, you were working in AI.
That's exactly right. I did some of the first developments in AI using technologies, existing technologies with AI.
Yeah, that's early.
The reality is that there's a couple different types of systems out there. There's frame-based, rule-based, neural networks, and so on, and they haven't changed that much. Our computing power is better, and
Mhm.
we've put some more algorithms together, and we have some more. But the reality is AI is going to provide us more ability to search a greater depth and greater breadth of potentials for products. It's not going to help us manufacture products any differently.
Mhm.
Products are still going to have to be made with, single-use disposables, stainless steel tanks, the proper technologies and so on associated with automation.
Mm.
What we have to be smarter about is in the early stages when we do a drug discovery is to work in a pilot plant that emulates what our factory is going to be like, our manufacturing facility. We spend too much time working with, we've got it in this little flask here, and what do we do for the next stage? Oh, we just do a little bit bigger flask. We got a bigger flask. Oh, we scaled up from 100 milliliters to 500 milliliters. Well, that's not, once you get to discovery, the
Mm.
jump to a pilot plant, an automated pilot plant, whether it's stainless steel or single-use disposable, is critical so that we understand scalability and we understand automation's impact. If you
Mm.
look back, turn the clock back to 2000, some of the monoclonal antibodies we produced, we thought we were going to get 1 milligram per milliliter as our titer. So what do? We built massive facilities, lots of trains, whatever. When we got it under industrial control, industrial equipment, good automation, whoops, we are 4 milligrams per milliliter. We didn't think that. So
Mm.
guess what? We don't need the X number of trains. We need X over 4 number of trains in our facility. And so we idled a lot of investment. Good pilot plant with scalable equipment, scalable vendors supplied material, scalable disposables, and those disposables are not necessarily just bioreactors.
Mhm.
They can be the TFF cartridges. They could be the chromatography columns. They could be whatever. But the key is scalability and reality. I've had development organizations tell me things like, "Oh, we're going to use centrifuges for separation." What kind of centrifuges? Oh, the test up. No, 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 we're not going to use that. Or we're going to
Mm.
use chloroform for extraction. No, you're not going to use chloroform in an industrial manufacturing environment.
Mm.
Let's adapt ourselves, our thinking, and our laboratory practices to where the endpoints scale is going to look like. And if the lab people aren't used to doing that, well, let's get them used to doing that. Because that
Mm.
is going to put us in a position to really understand where we're going to be, what the endpoint is going to look like, help us size the facility and get the investment correct.
What are your thoughts there, Herman, on that point about decarbonisation? Obviously, it's a big topic, but if we just take the bit about reducing energy usage, making for a more energy efficient facility, what have you seen working in that respect?
There's something that I did, if you look at what I did with a previous client in Raleigh, is we looked at the facility and said to ourselves, do we really need a WFI generator? Do we
Mhm.
really need a purified water system? Do we really need all the drains and all the other sorts of stuff associated with it? And so we built a facility that handles biologics, upstream, downstream, and fill finish, has no drains,
Mm.
has no water systems. And we did everything with single-use disposables. And if you need water, you're always going to need, you buy the water. So we bought the water in totes in different types of with already pre-sterilized disposables connected to it so that we could go ahead and so we did not have the liquid discharge that everybody else typically had. We didn't have to have a fired boiler system in there. We didn't need a CIP system. So
Mm.
by reducing, not having a CIP and a WFI and a purified water and all the drain connections, we changed our whole approach. Now, what did we do for getting rid of liquids? We bought a machine called an Actini machine from an Italian organization. And if whatever liquids we have, we would put it into that particular machine and it would sterilize it, keep it heated, and then it would discharge it.
Mm.
And it used a standard electrical hookup. So it didn't need a sterilizer or anything. So the philosophy should be if we want to try to is get as close to decarbonization, we want to get our carbon footprint down, is to think smart about our processes. Think smart about getting rid of a lot of the traditional systems in there. And again, I'm pushing us more towards single-use disposables in that particular vein. And then with the single-use disposables, there's things that can be done in terms of decontaminating them and shredding them and using them for different other applications.
Great, thank you. Herman, just finally, your son Eric, he's obviously followed your footsteps into pharma, and
yes, he has.
The pair of you write together and speak together on podcasts. Just wonder what it's like working with your son. I mean, do you have fun conversations at family gatherings? How does that work?
Oh, we do all the time.
Yeah.
First of all, I find it, and I believe he finds it immensely entertaining. We have a lot of fun doing it. When we get together at family outings, or we get together for whatever, we talk our own stuff, and then we kind of disappear because then we talk shop for a while. The interesting thing is my daughter-in-law, she's also in the pharmaceutical business. She's a pharmacokineticist for Janssen, J&J. So she'll join in the conversation. It's like, we're like three nerds, but it's an interesting life, and it's fun. Every once in a while he'll call me up and he'll say, hey dad, what do we do with this? What happened over here? Or he'll call and say, hey dad, you know what these jerks did? They put this into this and they expect, and the thing went boom. He said, oh, do you know this guy? Do you know that guy? So we have a lot of interaction from an industrial standpoint to try to improve each other's areas. And I've brought him into some of my projects, and I've helped him out in some of his areas. So we've had a lot of fun over the years doing this.
Experience the future of contract manufacturing at CDMO Live 2025. Join us at the World Trade Center Rotterdam on May the 7th and 8th, 2025 for an exclusive event that brings together the sharpest minds in biopharma outsourcing. For 2 days filled with market insights, best practices, and partnering. Early bird tickets are now available, so head to cdmolive.com to download the agenda and to book your ticket.